Hello, ladies and gentlemen. Thank you for joining us for our webinar. This time a very important topic for running 2D electrophoresis, namely the sample preparation part of it. According to our experiences, it is about 90% of the issues of insufficient or bad results in 2D electrophoresis are originating from issues with the sample preparation. And what happens is, this is happening mainly because you are dealing with very complex protein mixtures, where you have everything very hydrophobic, big, small proteins, sticky proteins, and enzymes, and so no wonder that it becomes so complicated. It's wishful thinking to extract 100% of all proteins and see them in the 2D gel. This is not possible. You have always to deal with some losses, but it is important that these losses are controlled. That's why that they are reproducible. So avoiding uncontrolled protein losses, you should, when you use the same sample preparation technique for the same sample, get the same result. That is absolutely impossible, otherwise you can forget your analysis. Another point is we want to preserve a protein mixture close to its in vivo state, so proteins should not modify each other because that can end up in a completely different pattern. So the first I show you is one which did not work at all. It was one of my collection of troubleshooting images, and there was a customer sending me that and asking me what was going wrong. And for sure I could tell him already it was a sample preparation, but maybe even other things have gone wrong. But you can't follow this up anymore. Complete disaster. A former colleague of mine, did a very nice experiment. The result you see here. He extracted Drosophila larva extract with 3 of the most used extraction methods published. So he looked in many papers and he applied the technique on the Drosophila larva extract. And you see immediately there are 3 completely different results. Just first look at the left one. Here you see that just homogenizing with 8 molar urea and CHUPS and DTT and so on not give a good result. And what you see immediately here is you see clouds in the acidic low molecular weight region, and this indicates that there was protease activities. So the proteases in the sample have become active after destroying the cell walls and have destroyed most of the proteins, and what you see are mostly peptides and digestive proteins. So this result is completely non-usable, and you find that I have seen many gels of this type, unfortunately. We know Drosophila larva extract contains a lot of proteases, but yeah. A second shows you much better image to like a 2D gel should more or less look like, without the vertical streaks, of course. But here the sample was homogenized in 2% SDS and boiled for 3 minutes. Now some of you would say, just a moment, you cannot first treat the sample with SDS, cover all the surfaces of the polypeptides to mask the isoelectric points and then run isoelectric focusing. This is impossible. You will not be able to focus the proteins on the IP. Yes, but you can. It's very important that you dilute out the SDS afterwards before you apply the sample onto the isoelectric focusing gel, and I will comment on this a little bit more later on. But what you see already now, this treatment with SDS is very useful to stop protease activities. Unfortunately, I have to tell you that does not last forever. So proteases are very stubborn molecules, and they can rearrange and start to to become active again after some time. So, but I will tell you another technique which is helpful also to kill protease activities forever. On the right, you see only a few spots. The procedure which was used here comes from the plant proteomics people, using TCA and acetone for precipitation. In plants, you do this to avoid building of protein polypeptide, polyphenol complexes, which are non-reversible anymore. But here, in this case, you see that the proteins have not come back into solution. So the Drosophila larva proteins are much more sensitive here to this precipitation technique. So, make it short, 3 different sample preparation techniques applied on the same sample give you completely different results. So you see the influence of the sample preparation technique. So what do we want to reach in sample preparation? First of all, we want to get and keep all protein solution, but always knowing 100% is not possible, but we try to come to this as close as possible. We need to stop enzyme activities, proteases, phosphatases and others, otherwise we get a completely different result, not indicating the in vivo state anymore. In some cases, pre-fractionation might be necessary or useful, but always bear in mind every step you apply in sample preparation additionally will lose proteins. You will always lose some parts of the proteins. But pre-fractionation might be quite useful. I come back to this point a little bit later. And we need to prevent distortions in the isoelectric focusing part, which are caused by contaminants. Isoelectric focusing is quite sensitive to salts and other contaminants, and we need to remove these contaminants without losing the proteins. It has long time not been known that even under high concentration of urea and detergent, reductant and carrion-phthalates, you will in some cases not be able to disrupt every protein complex. And so you need in some cases additional measures to disrupt the complexes. If you can't do that, the protein complexes are too large to go into a 2D gel, and so you lose the contents of these complexes. These proteins do not show up. Most of the isoelectric focusing instruments out in the labs are applying one electric field on all IPG strips in parallel. And if you have a few samples with a very high salt content, they will have very high conductivity, so they attract most of the current flow, and that means that these samples are separating in a different way than those with a lower conductivity. So it's very important that you achieve very equal conductivity of the samples, equal salt loads. Another point, already a little bit mentioned, very critical for sample preparation, modification of proteins are tissues, and like biopsy material, for instance. Tissue extracts are very difficult because when you open up the cell walls, you have immediately activities of all different proteins, and you modify your sample, or a sample becomes modified. The instrument you see here, the stabilizer system, has been developed in a company called Denator, and they also produce the system. And all this is an offspring of the University of Uppsala in Sweden, where they did a lot of protein and peptide research of brain proteins, for instance, with a huge problem. Of course, you can inactivate the enzymes by snap freezing in liquid nitrogen, for instance, and you can keep the sample frozen until you want to analyze it. The trouble, however, starts immediately when you thaw the sample. In very short time, the proteins will start to modify the sample, and then you can not get the in vivo state of the protein mixture anymore. So this system does another type of stabilization. It uses rapid conductive heating under vacuum. So as you see in the upper left picture here, the biopsy tissue is placed into the card-like plate here, which has a membrane on two sides. So you close it and apply it into this instrument, and it will immediately apply a vacuum on it. So the sample is immediately under vacuum, no oxygen influence anymore. And then a laser beam is applied to measure the size, the thickness, the width of the sample, and calculating the amount, the time of the conductive heating. So it's conductive heating, so you don't want to produce a steak, so to say, to heat more outside than inside, and you heat it to 96 degrees for a defined time fragment. And in this way, we have a standardized sample treatment. It stops degradation, and it will not start to become, the proteins will not start to become active again. You see here how that works. So you apply the energy by heating, and that means from the protein tertiary structure is modified, is stretched. The polypeptide chains are completely stretched, and when the sample cools down, it will rearrange into a three-dimensional structure, but when this is done under controlled way, not into the same way like it had been before. So it is aggregated, coagulated, but it is no longer active, but you can solubilize the proteins with urea, detergent, reductant, and so on. So I believe that for tissue extracts a very important development. Yeah, so the workflow in sample preparation, first you have to rub the cells. This is now what you use depends on the type of the sample, with tough cell walls or soft cell walls. We do not have now the time to go into the details here. Then you solubilize. You will see in the next slide how the solubilization, what you need, what reagents you need to solubilize as much proteins as possible. You need to protect against, again here, protease activities. It could be that you need pre-fractionation, but important, you might need to remove all these contaminants, nucleic acids, polysaccharides, lipids, salts. Insoluble material is centrifuged away because they can heavily disturb the first dimension. Solubilization, just as a rule of thumb, how the solution should look like. So the protein concentration should be about 5 milligram per milliliter, and that is now different for different sample types. Like even cell lysates are different in the protein content. So for lymphoid you need cell line, you need 50 million cells per milliliter. For HeLa you need 13 million cells per milliliter. It indicates you a little bit how the sample should look like optimally. Yeah, and you need high concentration of karyotrope. Usually you use 9 molar urea, or you can extract more of the hydrophobic proteins using a combination of 2 molar thiourea plus 7 molar urea. Unfortunately, this method can sometimes produce artifacts, so I would not use that as a standard method. I would always prefer to try this before and see if I can see more when I add the thiourea. It has been optimized that you need about 4% weight per volume detergent, and you need zwitterionic detergent, because what is always important, all the procedures, all reagents should be compatible with mass spectrometry after 2D electrophoresis. So therefore, non-ionic detergents are no longer used. Exclusively Zwitter-ionic detergents like CHAPS or other amidosulfobetaines like ASP-C7-B7O or ASP-12 or B14, or combination of a cocktail of all together of them are applied. Reductant, usually 1% DTT is used, or 2% volume per volume mercaptoethanol. Mercaptoethanol, of course, is not the preferred reagent, but in some cases you need it as a reductant when the tertiary structures are too heavily packed and too tightly packed, and then the smaller molecule can open that up. These two are usually preferred to TSEP. Some people use TSEP, tris-carboxyethyl phosphine, but the TSEP is not stable in the electric field, and you also add some conductivity to the sample by TSEP. So these are the standard here. And the carrier-amphalites, or also called IPG buffers, had been developed for forming pH gradients in isoelectric focusing, but for immobilized pH gradients separation you need them also because they support the solubility of the proteins heavily. That really helps. Secondly, the carrier-amphalites increase the conductivity of the immobilized pH gradients, because immobilized pH gradients alone have very low conductivity, and the result cannot be achieved without the carrier-amphalites. Sonication can really help, but you have to be careful not to heat the sample too much. You have to have a time and temperature control, otherwise you can create artifacts like carbonylation or other artifacts. Here you see another 2D gel which is not optimum, so to say. Yeah, that was during a course, and later on we found out what was the reason for this horizontal streaking. We were misunderstood, and instead of 4% CHAPS, only a tenth of it was used, 0.4%. And we did not know about this before, because usually when you use too low concentration of detergent, you are missing a lot of proteins. They are not going into solution. But here, obviously, they went into solution, but they show this horizontal streaking. Another experience that we collected. Never stops. Yeah, removal of nucleic acids might be really necessary with monocell material, or particularly with bacteria and yeast. And you can, for instance, use benzonase, but it's not so useful. We have a better reagent on the market since a short time, a cyanase nuclease, which is very fast. So you can get the full degradation of nucleic acids in a short time of a minute, and you don't see that anymore in the 2D image. That's one of the problems with other nucleases. You see the spot, but here the cyanase is removed with an inactivation resin, so you won't find it anymore. And you can keep the enzyme at room temperature. That makes it also much easier to use. Some samples contain a lot of salt, and then benzonases and others don't work anymore. But there is also a new product, the salt-active nuclease, which comes from marine bacteria, and they work even at half molar sodium chloride. And that is an area where others like benzonase would not work anymore. So keep that in mind. That may be very useful also in your 2D sample preparation. Also a few words on protease inhibitor cocktails. Here you see a list of cocktails which have been optimized for different sample types. Here you see bacterial, fungal, mammalian cells, plants, and so on. But it's highly recommended to use them during extraction of the proteins. But bear in mind, they are not helping forever. Yeah, the activity of some proteases can come back. I will come to that point again later on to completely irreversibly inhibit protease. Also, the same is the case for phosphatase activities, because you might want to watch phosphorylations, but you can't do that anymore if the proteins have been modified with phosphatases. Pre-fractionation. First of all, I would absolutely not recommend to use any protein chromatography technique when you are dealing with very highly complex protein mixtures, because large and sticky proteins can be lost on the chromatography material. This can lead as we don't want to have uncontrolled losses. Why would you like to fractionate according to subcellular components? Yeah, because there are many cases where you want to have topographic information. And yeah, just as an example, there are a number of proteins. Same protein can exist in different partitions of the cell. And when you would analyze just the complete lysis of a cell, when you would not be able to tell if the up or down regulation happens on the mitochondria or in the cytosol or where. How can you fractionate subcellular components, organelles? Yeah, the classical way is to use density gradient centrifugation, but very efficient is free flow electrophoresis, much more efficient even, or detergent fractionation kits, but the latter are, there are comments from some users that they're not so optimal. Should be a little bit careful with those. By affinity, but this is a critical point. You can do immunoprecipitation, but then you have only very low complex samples left over. Or if you try to remove protein complexes or abundant proteins with affinity chromatography, you have again a chromatography system first, and secondly sticky proteins like for instance albumin can also, if you want to remove the albumin from serum or plasma, you can also remove other proteins at the same time, sticking to the albumin. Blue native electrophoresis separates intact complexes, intact protein-protein complexes. And it has been used for so-called three-dimensional electrophoresis. So the complexes were cut out, the bands, the complex bands were cut out of the first gel and then submitted to a normal 2D electrophoresis. And so this three-dimensional electrophoresis showed, displayed very hydrophobic proteins which otherwise never showed up in a 2D gel. And there are possibilities to separate according to the charge, for instance between isoelectric membranes, but I would be careful here. Often proteins stick to these membranes, or again isoelectric focusing in free flow electrophoresis would be much better here. And that leads me to that slide. So if you want to apply free flow electrophoresis, there's only one protein in one company in the world is in Munich. This is the FFE service company, which does service. So they would separate your sample in their lab or in your lab, or you could buy the instrument from them. They would support you. And if you want to know much more, just contact ffeservice.com. You see here the latest development of the system. Here you have a thin stream of buffer. So you have no gel here. It's a gel-free system. You apply the sample at one point and you have a strong electric field. So in the strong electric field, proteins are going into this or that direction and they arrive always at the same point in the end. Here you have 96 tubing which end up in a microtiter plate to collect the different sample fractions. One example from organelle separation that was done in the University of Heidelberg, as you see here. First, there was a density gradient centrifugation of peroxisomes, and only by further treatment with free flow electrophoresis you see a good separation in the different subpopulations. Okay, if you're interested, talk to these people. One thing I announced already, but I should now explain a little bit more in detail, SDS in 2D sample preparation. It's mainly used for lipid-rich samples to remove the fatty stuff. It is used for tough cell walls like yeast and fungi. And there is another area where people deal with the troubles of having oligomers of proteins, for instance in serum, plasma, or antibody solutions. And here the people really found much better results by boiling the sample in SDS, in 2% SDS. But then, and this is very important here, you need to dilute out the SDS with 9-molar urea or combination of thiourea and urea plus 4% CHAPS, very important. And you should dilute it down to, rule of thumb, about 0.1% SDS. But what is even more important, this relation, this ratio, CHAPS to SDS should be higher than 8:1. That's important. Then, in the isoelectric focusing step, the SDS comes off from the proteins and you can achieve a separation according to the isoelectric points. As I already said, this inhibits proteolysis. Unfortunately, it's limited to low sample load because you are diluting the sample. And it can disturb the first dimension, mostly in that way that some proteins will have a shift to a more acidic isoelectric point because the SDS is not completely removed from these sticky proteins, for instance. So you see more and more papers where this sample preparation technique is applied. So again, sample preparation is very important, as I said several times already. It is needed to clean up from contaminants. It should break up complexes completely, as I said before, stop enzyme activities, and here really precipitation is very, very efficient. I would say the most efficient method. It is a one-step procedure, so to say, to clean up from all these different contaminants. In one, that would be very useful because you want to avoid many steps in sample preparation. Very important, this procedure is mass spec compatible. If you use other concentration techniques, for instance, with membranes, they often contain polyethylene glycol, and that can really interfere heavily with your mass spec analysis in the end. It irreversibly inhibits proteases, so that's the point I wanted to make all the time. This is the only method which is really inhibiting proteases completely irreversibly. It prevents and breaks up complexes. Really, this is an experience that we have made, very interesting. And for the Daiichi technique, different gel electrophoresis where you label proteins with a fluorophore prior to the 2D analysis, very useful because it removes endogenous peptides, so you label only the proteins and not the peptides. But bear in mind, there are only a few protein precipitation procedures which can be used, and they are listed here. One is useful here, TCA plus acetone, coming from the plant researchers, but it loses some of the acidic proteins, so be careful with acidic proteins would be lost. But it for basic protein analysis, very useful. Chloroform methanol by Wessel and Flügge is very often used in many, many protein labs, proteomics labs, because it is very useful. You have a very good resolubilization of the proteins coming back into solution. And another one is a combination of TCA, a detergent, usually that's deoxycholate, and acetone, but these are commercial 2D cleanup kits. It's not they don't tell you exactly what's in it and how it's combined to each other. They're also very good and very useful. Sometimes even the chloroform and methanol works better. In other cases, for other samples, this procedure works better. But these are the precipitation procedures we can recommend. Two examples. One also from a customer. On the left, you see, was a sample extracted with urea, detergent, reducted, carrier phthalates, and the result was absolutely no separation in the first dimension. But when they used precipitation, chloroform, and methanol procedure, same sample. You see now that's almost perfect 2D gel. Another case we saw during a course that we ran in Malaysia, in Kuala Lumpur. We had a very strange sample received from the customers, rat tongue tissue, and no separation in the first dimension. But using commercial sample preparation kit, you see suddenly all the proteins showed up and we got a normal 2D. So that two examples showed you how important it might be to break up complexes and to clean up your sample. But a critical point is always left over, namely, do I really get all my proteins resolubilized? And there you need to take care. There are a few points which are really very important. First of all, a pellet must never become completely dry. It is very dangerous when you use these vacuum centrifuges, speed vac and so on, and don't care. When a protein pellet has become completely dry, it cannot, the proteins don't go back into solution. The structure, so to say, collapsed. You can pipe it repeatedly, lysis solution over the pellet, but you should, very important, not vortex. These vortex systems which are on these lab benches are very efficient, but here they are too efficient because they are causing foaming of the detergent. Remember, you have about 4% detergent in your sample, and then the proteins are foaming and they don't go back into solution. Don't vortex, but you can finger snapping them, you can shake them a little slightly, but not vortex. Often you just need to place your Eppendorf tube somewhere on the bench for a few hours and wait, and you can see after a few hours the protein pellets have been completely dissolved, proteins have gone back into solution. But also here, don't vortex. You can carefully sonicate to support the resolubilization, but as already said before, avoid heating of the sample. Use time and temperature control here. You can grind up the pellet to increase the surface with a little sticks, for instance. Then proteins go easier into solution. You can freeze the pellet with lysis solution. That causes crystallization of urea and increasing the surfaces also. And if all this does not work, if there's still some precipitate left, you can always go also to the SDS procedure as mentioned before, but then please, you have to dilute out the SDS down to this value 0.1%, and the most important rule of thumb here again is the CHUPS to SDS ratio must be above 8:1. Just a few special cases where should you do what. Bacteria have a high nucleic acid content, so you have to remove the nucleic acids, otherwise you have a horizontal streaking in the acidic part of the gel. Yeasts and fungi have very tough cell walls. Here a lot of people are using the SDS procedure. Cell culture is dangerous because you have to wash the cell culture material off the surfaces of the cells, and usually people are using PBS, phosphate buffered saline, and this has to be completely removed from the surface, and this is not always so easy. So then we recommend to do an additional washing with sucrose 250 millimolar in 10 millimolar tris. This does not destroy the cells, but it removes the salts, and you have no disturbing salt load on your sample. And finally, plant tissues, very tricky sometimes. There are several papers around, especially dealing with plant protein extraction. You have to avoid phenol protein complexes. The last point, which usually you would say would not belong anymore to the sample preparation, but now it starts to belong to it, more or less. The traditional detection methods are all using post-staining, like Kumasi. Silver staining is many steps, very sensitive, but many steps. Still, also the fluorescent staining methods require several steps, and what turned out to be very useful is to pre-label the sample prior to the first dimension, like with SERVA lightning red, and then you can directly after the 2D run image the picture. You can fix afterwards, or you can use continuous blotting and other detection techniques. So I think that there is a trend going to that, and so it more or less belongs to now to the sample preparation. And here you see one result with the SERVA lightning red. Gives very nice result, makes the entire process much easier and better reproducible. Okay, that was what I wanted to tell you today, and I thank you for your attention, and I'm now prepared to answer your question, and bye-bye.